All right, good morning, everyone. Today I want to present our recent findings on individual differences in executive control. And the main rationale behind all this work is that we're looking at, individual differences and try to explain them from a molecular genetic perspective. So this will be about genes influencing executive control. And as a theoretical background, we're using the working memory model by Alan Baddeley. I think you will be all at least a little bit familiar with this concept. The main idea behind it is that there are like multiple different local processes in the brain processing more or less bottom-up information, and that there is one central executive processing unit which is more or less in charge to control all these other processes in the brain. And Baddeley himself, when he put up this model, he focused more or less on all the different components, and he never put any effort into decomposing the central executive. And so this was the task for other guys. And this is a pretty recent latent variable approach where people try to assess executive control and try to look whether there's like an individual factor underlying like everything, kind of similar to the G factor of intelligence, or if there are different subfacets of executive control. And the main finding was that there is a single factor underlying executive control, but that there are also three different factors that influence executive control on the level just like one below. And this is shifting, updating, and inhibition. And in my talk, I will focus on shifting and on inhibition, and I kind of neglect updating. And shifting, I think the best way to think about shifting is to think about your everyday work in the office. You're just like working on multiple tasks all the time. So you're pretty much likely to write a paper when the phone rings, pick up the phone, answer the phone, put the receiver down, work on the paper again, look at somebody who's knocking at your door, whatever. And every time you perform on a different task, you have to switch between one task to the other. And even if you might find this very easy, if you are in an experimental laboratory and repeatedly switch between tasks, you can always measure a little bit of a performance decrease whenever a task switch occurs. Performance decrease means when you measure, just like correct responses, you see like an increase in errors, and you also see an increase in reaction time. So there are like a little bit of cognitive processes going on that help you to shift your attention between different tasks. And the main explanation for that is some sort of task set reconfiguration. And a task set is a concept from the cognitive literature, and the main idea behind this task sets is that there are mental representation in our brain, in our memory, in our cognitive system, and that these sets just constrain your behavior in ongoing tasks. So in this task sets, representations in long-term memory, which are activated into working memory, are all the information you need to perform on a specific task. And whenever you have to switch between two tasks, you have to deactivate the current task set to start the new task set in order to perform on the second task. And I think the best way to think about this is just like imagine how you drive a manual car. When you have to switch gears, you first have to ungear just in order to put the next gear on. And this is like the idea behind task set reconfiguration. Next thing I wanted to talk about is inhibition, the second executive control function. And inhibition is an active suppression of information or even a cognitive representation. That is, whenever a specific information, a specific representation becomes irrelevant, then like an inhibitory control mechanism is activated that suppresses this information so that it cannot interfere with the actual ongoing task. And this is actually a thing you can think when you think about this task sets. You can imagine that the task set is no longer relevant because you have to abandon this task in order to switch to another one. That this task set, which is irrelevant now, gets inhibited. And that's the main thing I'm going to talk about now, about the inhibition of task sets. And I will introduce a task I'm using, or the task which was used to study inhibitory control during task switching. And this is a stimulus people see during the task. So there are four rectangles, they're all blue, but they kind of differ. And now I don't have the animated version of my task, so please just imagine that the one on the left down side is oscillating in the horizontal plane, right? It's moving constantly. And there are three different tasks people can perform on this, and the relevant task in the current trial is always indicated by a small verbal cue in the middle. So in this case, it's an M. This is the movement task, and the participant has to point out which of the four rectangles is odd on the movement dimension, which means that if this one on the lower back, the lower left is moving, then people would have to point out, well, this is moving. This is the movement task. Another task would be the orientation task. It's indicated by an O for orientation, and participants have to point out which stimulus is deviating on the orientation dimension. And as you can see, the one on the top right, it's tilted 45 degrees clockwise, and so this is like the deviating stimulus here. And the third task would be the size task, and participants have to indicate which one deviates in size, which is either smaller or larger than the other rectangles, and this is the one in the top right. So we have an identical stimulus on each experimental trial, but 3 different tasks associated with them, and all these tasks are indicated by a small letter. And what we do then is we look at task performance on the stimuli over time, and we kind of look at the performance as a function of the stimulus which was on before, and this is the rationale behind it. So we have like the trial N, the lower one, that is the current trial that the participant performs on right now. Then we have trial N-1, that was the last trial immediately before that, and N-2, that's 2 steps back. And when we think about this task set concept, when we perform on the movement task, we have to activate the task set movement. Then, when we perform on the orientation task, we have to activate the task set orientation. And when we switch from movement to orientation, we inhibit the task set movement in order to perform on the task set orientation. And when we then switch from orientation to another task, we have to inhibit the task set orientation. And now the thing is that we compare 2 different conditions, and these are the one in the M trial, at the lower one. So the first possibility is you perform on the movement task, then switch to the orientation task, then switch again to the shape size task. And this is what we label the control condition, because we have 3 different task switches, but we switch from task 1 to task 2 to a third task, the neutral one. And we compare it with this condition where we switch back to the movement task. So the idea here is to perform on the movement task, switch to orientation, and then switch back to movement. And the idea behind the inhibitory control is that when you switch from movement to orientation in the first place, you inhibit movement, and this inhibition is still present at the current trial because it slowly decays over time. So what we should find here is that the movement task, which needs to be performed right here, is still some sort of inhibited, and it's pretty hard to overcome this inhibition and to start it again. And as you can see in these empirical data by Meyer and Kiel, when you have to perform on this inhibition condition right here, that is, you're switching back to a recently inhibited task, performance measures decrease. That means an increase in reaction time compared to this control condition. Right. And what we did in our present study, we thought to look at individual differences in the strength of this inhibition effect and try to explain it from a molecular genetics perspective. And the main rationale behind this is this executive control is highly heritable, and with highly heritable, I mean 99%. That is like almost the entire variation in executive control can be attributed to genetic factors. And this is true for the common factor, but this is also pretty much true for the inhibition factor, which is heritable to 99% as well. And the idea is now, can we explain, or can we look at this endophenotype of executive control, this backward inhibition effect during task switching, and explain individual differences? And first of all, we need a good candidate gene, because there are so many genes and so many genes just expressed in the brain. So we need a clear-cut hypothesis. And we focus on one gene, the DRD2 gene. That's the one Nora was presenting about earlier. It codes for the dopamine D2 receptor. And these receptors, the dopamine D2 receptors, are thought to mediate executive control. So that's the first reason we focus on this gene. And it was shown that for this mutation, for this polymorphism, the TAC1A polymorphism, that the A1 plus variant is associated with a decrease in the receptor density in the human striatum. So this is like a functional variant, and we can actually compare participants carrying this A1 plus variant with those who don't. And there is one study from colleagues of ours about task switching and the DRD2 gene, and they were able to show in a regular task switching design without any inhibitory control component involved, that participants who carried this A1 plus genotype with a reduced receptor density were actually better in task switching. So we have an already a link to task switching, and now we're looking at inhibitory control during task switching. We tested 100 participants with the task I just showed you, and we genotyped them by means of real-time PCR on this nice machine for the DRD2 TAC1A polymorphism. And as you can see from the genotype frequencies, the A1 genotype, that means you carry this A1 variant on both chromosomes, pretty rare in Europeans. So we grouped them together. So we compare 31 subjects carrying the A1 allele on at least one chromosome with 65 who don't carry this variant. The behavioral protocol was the odd man out task that I just showed you. So there was always a verbal cue indicating that movement dimension is relevant in the next trial. It was shown for 100 milliseconds, so pretty briefly. Then the stimulus popped up. The participants had to respond on a response pad, and then they had like a pretty brief interstimulus interval. In total, participants performed 200 trials and on all 3 tasks and switched the task on each trial. So these are the results from the group analysis. So no individual differences yet, just like the whole group, all 100 people. And you see on the left the non-repetition condition, that is the control condition. On the right side, the repetition condition, that is the inhibition condition. And these are reaction time data. And you see a pretty significant increase in reaction time whenever participants switch back to recently inhibited task. This is, first of all, the replication of the literature on the group level. And then we focus on individual differences according to the genotype. And now on the left side, you see those guys carrying this A1 plus genotype with a reduced receptor density. On the right-hand side, you see the A1 minus carriers, so the 65 participants who don't carry this allelic variant. You see the reaction times on the y-axis, and the red bars are the repetition condition, where the inhibition should occur, and the black bars are the control conditions. And first of all, you see this increase on repetition trials in both groups. So we have the backward inhibition effect in both groups. But it's somewhat stronger, and this is qualified by a significant interaction between task condition and genotype for A1 plus carriers. That is, those guys have a pretty much stronger backward inhibition effect than those people who don't carry this genetic variant. All right, just like an attempt to explain these results, or what does it mean, what we found. First of all, we were able to replicate the backward inhibition effect in a pretty large sample. With pretty large, I mean 100 people. And we were successfully able to associate the DRD2 gene with the backward inhibition effect, and we were able to explain 6% of the variants in this phenotype. So to sum up, the carriers of the A1 plus variant, first of all, they show the reduced receptor density. They're better at task switching, and now we were able to show that they have a larger backward inhibition effect. And to some extent, this replicates the results by Stelzel et al. With the task switching thing. And for those of you who are familiar with molecular genetics of cognition, it's pretty hard to get a replication in this field. So this is actually kind of cool that we were able to replicate results from another work group. And we kind of see backward inhibition as an endophenotype of task switching and an executive control. And one aim of ours is just like to disentangle the specific molecular influence on cognition by closely manipulating experimental tasks. So just like to explain what genes do by looking at experimental psychology. And the ultimate goal would be to identify, of course, all the genes contributing to this highly habitual trait executive control. So what will be next? First thing I'm going to do is I want to look at other inhibitory phenomena during task switching. Right now I'm collecting data, and just like to spoil you a little bit, they're looking pretty good, and I hope I can present it next time. And then we kind of assess the role of other genes, especially the COM genotype. And what's kind of interesting in the inhibition literature is that usually if you use other inhibitory tasks, such as the anti-saccade task, the stop signal task, you usually find an effect of the COM15 MET polymorphism on inhibition. And we tested for that, of course, as well, and we didn't find such an effect, but we found the effect of the DRD2 gene. So it would be nice to show like this double dissociation in one data set, so that would be next as well. Then to carry all this into a neuroimaging environment, to look for real-life approaches, you know, just like the office kind of thing. And of course, to look at the clear relationship between shifting and inhibition, because we're looking at inhibition during shifting, and these are clearly separable factors. So the question is, how do they interplay with each other? So that's the thing I'm going to do, and hopefully get some suggestions from you guys. First of all, I want to thank my colleagues who helped me to work on the thing, Martin, Christian, and Nora, and Thomas. And just in case you want to read anything up, there's a paper out on this data, and I'm more than happy to send it to you if this would be of any interest to you. Thanks.